Welcome back. Cell, the unit of life, covers the cell theory and the internal structure of prokaryotic and eukaryotic cells, the foundation for nearly every later biology chapter. In this video, we'll cover cell theory, the differences between prokaryotic and eukaryotic cells, and the key organelles found inside a eukaryotic cell. Let's begin. Cell theory states three foundational principles. All living organisms are composed of cells. The cell is the basic structural and functional unit of life, and all cells arise only from pre-existing cells. This last principle specifically rules out the idea of spontaneous generation or new cells from non-living matter. Cells are broadly classified into two fundamental types. Prokaryotic cells, found in bacteria and archaea, lack a true, membrane-bound nucleus. Their genetic material lies free in cytoplasm in a region called the nucleoid, and they generally lack most membrane-bound organelles altogether. Eukaryotic cells, found in plants, animals, fungi, and protists, have a true, membrane-bound nucleus enclosing their genetic material, along with a variety of specialized, membrane-bound organelles that divide the cell into functionally distinct compartments. This organelle-based compartmentalization is exactly what allows eukaryotic cells to carry out many more complex, specialized biochemical processes simultaneously without those processes interfering with each other. Every cell is bounded by a plasma membrane, described by the fluid mosaic model, which pictures the membrane as a flexible phospholipid bilayer, with proteins embedded throughout, some spanning the entire membrane and others floating on just one side. And this fluid dynamic structure allows the membrane to selectively control what enters and leaves the cell, making it selectively permeable. Small nonpolar or uncharged molecules can generally diffuse directly across the lipid bilayer, but larger, or charged polar molecules need specific transport proteins to cross, either through passive facilitated diffusion, which requires no energy, or through active transport, which requires ATP, to move substances against their concentration gradient. Plant cells, unlike animal cells, have an additional rigid layer outside the plasma membrane, the cell wall, made primarily of cellulose, which provides mechanical strength and structural support, protects against mechanical stress and pathogen entry, and importantly prevents the cell from bursting when it absorbs excess water. A critical difference from animal cells, which lack a cell wall entirely and can indeed burst under similar osmotic conditions. The nucleus is the control center of the eukaryotic cell, bounded by a double-membraned nuclear envelope, studded with nuclear pores that regulate the exchange of molecules like RNA and proteins between the nucleus and cytoplasm, and it houses the cell's chromatin, DNA loosely bound to proteins, which condenses into visible chromosomes during cell division. Several organelles form a functionally connected endomembrane system. The endoplasmic reticulum comes in two forms: rough ER, studded with ribosomes and responsible for synthesizing proteins destined for secretion or membranes, and smooth ER, lacking ribosomes, responsible for lipid synthesis and detoxification. The Golgi apparatus receives proteins and lipids from the ER and modifies, sorts, and packages them into vesicles for delivery to their correct final destination, often described as the cell's packaging and dispatch center. Lysosomes, membrane-bound sacs of digestive enzymes produced by the Golgi apparatus, break down waste material, worn-out organelles, and material taken in from outside the cell, earning them the nickname suicide bags, since their rupture can trigger the controlled self-destruction of the entire cell. Mitochondria are the cell's powerhouses, generating most of the cell's ATP through aerobic respiration, and they have a distinctive double membrane structure, with the inner membrane highly folded into structures called cristae that dramatically increase surface area for the biochemical reactions of respiration. Chloroplasts, found only in plant cells and algae, similarly have a double membrane and are the site of photosynthesis, containing internal stacks of membranous sacs called grana, connected by structures called lamellae, embedded in a fluid matrix called the stroma. Notably, both mitochondria and chloroplasts contain their own small circular DNA and ribosomes, and can divide independently of the cell, which strongly supports the endosymbiotic theory, the idea that both organelles originated as free-living prokaryotic organisms, engulfed by an ancestral eukaryotic cell long ago. The cytoskeleton, a network of protein filaments including microtubules and microfilaments, running throughout the cytoplasm, provides mechanical support, maintains cell shape, and enables both intracellular transport of organelles and, in some cells, whole cell movement. Let's recap. Cell theory establishes the cell as life's basic unit arising only from pre-existing cells, and cells split into prokaryotic, no true nucleus, and eukaryotic, with a true nucleus and organelles. The fluid mosaic plasma membrane controls what enters and leaves, while the plant cell wall adds structural support. The nucleus controls the cell, and the endomembrane system, Golgi apparatus and lysosomes, handles synthesis, processing, and waste disposal. And mitochondria and chloroplasts, both bearing their own DNA supporting endosymbiotic theory, generate ATP and carry out photosynthesis respectively, while the cytoskeleton provides shape and enables transport. That's cell, the unit of life. This chapter's organelle functions come up constantly in later chapters, so lock them in now. See you in the next video.